Hi, welcome to the Novogene Introduction to Non-Coding RNA-seq Analysis Contents. If we can just give it a couple of minutes, as we've got a lot of people joining at the moment. Bear with us, we'll start in about 2 minutes. Hi, welcome to the latest Novogene webinar, an introduction to non-coding RNA-seq analysis contents. I'm John Cafferkey, Marketing Manager here at Novogene Europe. This is the 7th in our lockdown webinar series. We complete the series on the 23rd of July with solutions for microbial genomics, and then we take a short break over August. We will return with a full program in September. We'll be emailing out full details soon. A bit of housekeeping. The webinar will last 1 hour with approximately 10 minutes question and answer session at the end. Please submit your questions via the question and answer chat functionality you will see in the middle at the bottom of your screen. We'll answer as many questions as we can. If we can't answer your questions live, we will get back to you by email after the webinar. I'd like to introduce you to your host for today, Caterina. Dr. Caterina Abaldi did her studies in medical molecular biotechnology in Italy and got a PhD in human pathology and molecular medicine at Sapienza University of Rome. She was a senior scientist in a genetic testing company for rare disease, was responsible for somatic variant detection in cancer. Since July 2019, she's been the Novogene Regional Account Manager for Brandenburg and Mecklenburg areas in Germany. Caterina, over to you.
Thank you, John, and good morning to everyone. This is Caterina Baldi, and today I will give you a general introduction of Novogene Europe before the webinar will start. So Novogene was founded in 2011 and has been growing rapidly over the past few years. As you can see here in the picture, our work is focused to improve life science research, clinical diagnostic, and human healthcare, encompassing all applications of biology and life science. We have strong scientific expertise and experience with 29 NGS-related patents and as well over 5,000 research papers, including publication in first-tier journals such as Cell, Nature, and Science. Novogene uses the best and proven technologies from Illumina Nova 6000, HiSeq, Oxford Nanopore, Ion Torrent, and PacBio SQL platform. Over the years, our state-of-art NGS lab have optimized various NGS protocols to maximize scientific outcome for your research. We also own the largest sequencing capacity in the world with an annual throughput of 208,000 human whole genome sequencing samples. Our large assembly of NGS system enable us to run projects of any size, including those that require massive amounts of sequencing and data analysis. Novogene has a strong global presence with established operations and laboratories in China, in the United States, United Kingdom, Singapore, Hong Kong, Japan, and the new one in Netherlands. We serve more than 30,000 customers in more than 60 countries globally, and have an extensive collaboration with many world-class university, research institutes, hospital, biotech, and pharmaceutical companies. Novogene deliver unsurpassed data quality to support our customer research goals. We have a range of NGS-based service for human, plant, animal, and microbial DNA and RNA analysis. Today, our webinar will be focused on non-coding RNA sequencing, as you know. Novogene Europe sales team is present in most of the European countries. And as you can see in this map, we are ready to support your project closely to you, discussing in meetings and find the best solution for your research needs. At the end of this webinar, during the QA session, you will see a slide with all of our faces and contact details, so feel free to contact us. Our sales team, is big. We are 21 regional account manager and very international with 15 languages spoken. So, and we can help you achieve your research goal. How we can do? First of all, giving our best informatic expertise and strict adherence to quality control, and this results in the highest quality data. Giving to our customer publication ready data if decide to add our bioinformatic analysis, selecting the most effective platform and strategies for our customer needs, and completing your project in a very short turnaround time due to our high experience and professionalism and our NGS capacity. So this is our sales team. So feel free to find your sales manager. And I would like now to give a brief presentation of Dr. Wei Li, who will present the webinar today. She did her study at Clemson University, US, and got a PhD working on cotton transcriptome analysis. She started her career in Novogene as a senior bioinformatic engineer, and then promoted to a senior product manager for Novogene Europe. I would like to now stop sharing and give the microphone to Wei, and thank you for your attention and enjoy the webinar.
Thank you, Katerina, for your introduction. And now I'm going to share my slides. Can you all see my slides?
Yes, sure. Thank you.
Okay, great. Thank you. So again, thank you for joining us in the webinar. So today I will give you a brief introduction of non-coding RNA-seq analysis contents provided by Novogene. In this webinar, there will be 3 parts. First, we will go over the general non-coding RNA-seq services, and then we will use the LincRNA analysis pipeline as a demo to show you the detailed steps. And in the last part, we will provide you some other solutions for the non-coding RNAs. First, as you can see from this picture on the right, so basically, ribosome RNA is actually taking a huge proportion of our total RNA. Well, in the other small part showing on the top, you will see a small proportion of messenger RNAs and even little percentage of the long non-coding or other non-coding RNAs. Even though they are taking a very little proportion by the masses, but you will see their potential significance or importance in their regulation of the gene expression, as indicated in on the left. So those non-coding RNAs can either interact with our messenger RNAs or compete with each other to get involved in the regulation of the gene expression. And to take care of each of our non-coding RNA-seq service or projects, we will follow this workflow. So first, we will start with the total RNA preparation. You can either extract the RNA in your lab, and we will have some recommendations for the extraction method. Or if you request, we can also offer you the extraction services. So you just need to deliver your sample to the UK lab, and we will take care from there. And after your total RNA is ready, we will move on to the following steps, including sample QC, library preparation, library QC, sequencing, data QC, and bioinformatic analysis if you need. So for the non-coding RNAs, based on their different configurations, we will have different library to target them. For messenger RNA, it will be quite efficient to just use the poly-A arrangements or some other methods. But for the non-coding RNA, as not all of them have the poly-A tails, so we need to choose an alternative. So that will be the ribosome RNA removal. If you recall the pie chart I showed you in the previous slide, you will see there will be a very huge portion of the ribosome RNA. So that's how we need to enrich those non-coding RNAs, including LincRNA or circRNAs. And to accurately quantify them in our analysis, we need their strand information. So we will also construct the directional library for both of them. While for the small RNA, as they have a very special small size, so we will just use the size selection during the library preparation to effectively enrich them and move on to the sequencing. So except those library options, you can also prepare your own library in your lab. We also accept pre-made libraries. But before you send it out, please double check your library structure with us. So we need to confirm your library is compatible with our platforms. And now let's go to the second part to see some analysis details for the LincRNA. Before we move forward, let's check how or what kind of question we try to answer by using RNA-seq. So as I'm showing here, actually majority of our cells are having exactly the same genomic DNAs, but why not all of them are functioning the same? So such as we have some muscle cells or we have some nerve cells, they function differently. And one of the major reasons would be their transcriptome differences within different cells. So they may either express different transcripts, like through the alternative splicing, or even though they are expressing exactly the same transcript, maybe they will have different abundance in different cells. So basically in the RNA-seq analysis, we care two questions the most. First is the annotation of our transcripts, like what transcript is expressing in my cells. And the other one will be the quantification. We want to quantify the transcripts in different samples or different cells. So that's how we carry out the messenger RNA analysis in general. So we collect our data and then we process our data, like we just pick up the clean data for the future analysis. And after mapping, you will see two major parts. First will be the left branch that includes the gene quantification. It will help us to address the abundance question. And the other one will be the structure analysis. This will help us to identify which transcript is there in our samples. So is that enough for our non-coding RNA-seq analysis? So if you check the figure showing on the right, so those non-coding RNAs will not directly involve in the protein coding. So they cannot directly to produce those proteins as the messenger RNA do. So basically, they will either interact with some other moleculars such as DNAs or proteins or even other transcripts. So that indicates their indirectly involvement of the protein coding, which is the regulation of the gene expression. So to address that question, our third question in the non-coding RNA-seq analysis, we will add an additional step, which is the target prediction in the non-coding RNA-seq analysis. So here, we will just use our LincRNA to show you the details, like how we can achieve or how we can answer those three questions successfully. So in our LincRNA analysis or service, so basically our long non-coding RNA will be the non-coding RNA longer than 200 nucleotides. And if you want to run the analysis by us, so we always need a reference genome available and together with the complete annotation. And in this service carried by Novogene, you can collect the information for both messenger RNAs and long non-coding RNAs. So I will talk those information later in the analysis part. And again, for the library preparation, we will prepare the strand-specific library with ribosome RNA removal. And here will be some data recommendations. So you may adjust the data amount request, request based on your projects. And below will be some details for the sample requirements. So you don't have to worry if you cannot take all those numbers right now. So we will share the slides after the presentation, and we will also have our local representative available all the time for you to get those details. So here will be our LincRNA-seq analysis pipeline. And again, from the very beginning, that will be our raw data collection. After we get our raw data from the sequencer, we want to check our data if they are good enough for us to use. But there are some two specific steps I want to emphasize first at here. So after the mapping, the same step as the messenger RNA analysis, we will identify the long non-coding RNAs. So this is because when we collect or enrich the long non-coding RNAs in the library preparation, we can, from the pie chart again, we can recall that we also collect some messenger RNAs or some other non-coding RNAs. We need to use some criteria to filter out those non-LincRNAs, or we need to identify the real LincRNAs in our samples. And after that, we will follow similar pathways to quantify those transcripts and perform the differential expression analysis, and then will be some enrichment analysis and some other functional analysis. Again, for the long non-coding RNA or all of other non-coding RNA analysis, we will also include the target prediction in our pipeline. So now let's go through each step in a little bit of details. First, again, from the raw data QC. So after you got your raw data, maybe not all of them are good enough. So we will check the raw data or assess the raw data quality by showing you some figures. Here I show the sequencing quality distribution as an example. So you will see how good your data are. And for the Novogene service, we always guarantee some good data quality. Such as for some of our RNA-seq projects, we guarantee the Q30 higher than 80%, which exceeds Illumina official guarantee. And after we get our raw data, we also classify your data into different categories, such as some of your reads may have very smaller insert size, and in those reads, you may see the adapter sequences as well. So those reads will be our reads containing adapters. And some of the reads may have very low quality or even with a lot of uncertainties, a lot of N's. So we will also classify them based on our categories. And the last portion will be the clean data or clean reads. So for the projects without bioinformatic analysis, we will deliver all raw data, and this data assessment or data QC report to you, showing you the distribution of your data. If you request the bioinformatic analysis with us, so we will only use the clean data to perform the following steps. So now we have our clean data ready, and now we will move on. Next step will be the mapping. So we map all the short reads against our reference genome. So the reference genome can either come from those listed browsers, genome browsers, or we will also provide a list of the reference genomes which are already available in Novogene clusters or analyzing systems. So you can choose based on the list, or either provide us your preferred reference genomes. And through mapping, we can tell where our reads mapped to, and if this region has been annotated with any potential biological functions. So that will be the most significance of the mapping step. And in our report or results, we will show their mapping abundance or distribution along the different chromosomes. All of the mapping results will be saved in the BAM format or BAM files. So you can load your reference genome and the BAM files or mapping results in the IGV software, and to check their counts, read counts, or which region they have been mapped to. So those will be the software we recommend you to visualize your mapping result. And after the mapping, as I mentioned, we need to identify our real lncRNAs. So we will use some filtering criteria. So first, we will check their exome number and expression level to filter out those false positives. And after that, we check their size. As we know, long non-coding RNAs will be the non-coding transcripts longer than 200 nucleotides. And then we also identify or mark if this transcript is already an annotated one or unannotated one. So this one or this result will be used for our next step, the Novogene prediction. And the last category or the last step we will perform will be the coding potential assessment. We'll use 3 software to predict the coding potentials for our transcripts. And finally, we got the LincRNA identified, and then we will classify them into different groups or different categories based on their feature or annotations. And after the long non-coding identification, or before the quantification, we will add another step, which is the Novogene prediction. As for some of the genes has not been fully annotated in the reference genome, so once we got any Novogene predict, we will add those annotation information in your result release package, and then it will enrich or better annotate your reference genome as well. So here I just show an example to let you get an overview like how those annotations will be like. And with our LincRNA identified and Novogene identified, we will move on to the quantification. So in this step, we will quantify how many reads has been mapped to each of my gene. However, my read counts is actually affected by a lot of reasons, and those 2 major reasons will include the total read counts, like how many reads I sequenced for each of my samples. Maybe in sample 1, you got 10 millions of reads, but in sample 2, you only got 100 reads. So those will affect your read counts significantly. And the other factor will be the gene length. It's quite possible you got much more mapped reads in your longer genes if you compare that against your shorter genes. So to compare or easily compare your data across samples or across genes within the same sample, we need to normalize our raw read counts against the 2 factors. That will be the total read counts and gene length. So the FPKM, fragments per kilobase per million, is the commonly used normalization method for RNA-seq. So you just need to normalize your fragments counts against the gene length and the total read counts. And in the result part, you will see the FPKM value for your lncRNA and your messenger RNA. As I mentioned before, we will include the information in the LincRNA service for both lncRNA and messenger RNA. And it's quite obvious that you will see much lower expression level in the lncRNA transcripts if you compare that to the messenger RNAs. That actually making sense, and that's also the other reason we recommend you to sequence more in the lncRNA service or lncRNA projects, because it got really low abundance in their expression levels. And we also include different formats of figures to show the distribution of their FPKMs, such as the curve, the density curve, the box plot, and the violin plot. So again, we will include that for both LincRNA and the messenger RNA. And the next important step will be the target prediction. As we mentioned that the LincRNA will involve a lot in the regulation of gene expression. And to predict the targets, we will first assess the co-expression relationship between the LincRNA and the messenger RNA using the Pearson correlation value. So whenever the PC or the Pearson correlation value is higher than 0.7 and the p-value is lower than 0.05, we identify this as a co-expression or a potential target for the LincRNA. So there will be two different regulatory relationships between LincRNA and the messenger RNA we will include in the result. First will be the cis-regulatory. If your LincRNA and messenger RNA locates on the same chromosome and their distance is within 30 kilobases, so that will be classified as cis-regulatory or cis-regulatory relationship, or in this category. And anything out of this range, like they locate on different chromosomes, or my messenger RNA is super far from my LincRNA location, so that will be classified into our trans-regulatory relationship. And in this part, we focus on the transcription factor identification or target identification. And with the quantification information, now we can move on to our differential expression analysis. You can define your comparison group, like which sample I want to compare between in your BI form, showing like this. And once we have the comparison group, we will compare the expression level between those two samples or two groups. We always include the volcano plot and heat map to show the differential expression analysis result. So volcano plot is something shown on the left. And based on your defined group, we will plot each gene in this volcano plot. And the dot with positive and negative log2 change is actually indicating upregulation and downregulations respectively. We also label out the cutoff of the significance. Whenever the genes with the lower p-adjusted value will be on the above, as the y-axis is showing the negative log10 p-adjusted value. So those genes above the dashed line will be the significantly differentially expressed genes or transcripts. So from this figure, we can tell the red and the green are showing the significantly upregulated and downregulated transcripts respectively. So this is how volcano plot can deliver the information. And the other figure we will include will be the heat map. So in the heat map, we will cluster all the differentially expressed transcripts based on their expression profiles. That means if they have very similar expression behavior or profiles within my experimental design, they will be clustered together. So this will be very useful for us to predict the function of some or some poorly annotated genes, as the genes with a similar expression profile will be clustered together, indicating they respond similarly to the experimental design. So that will be very useful for us to make the functional prediction of those poorly studied genes. So again, for the volcano plot and the heat map, we will also include that for both LincRNA and messenger RNA. And next step will be the enrichment analysis. In this step, we want to know which either gene function or biological pathway has been significantly enriched for my differentially expressed messenger RNA or differentially expressed LincRNA targets. So in this one, we will find out, or we will plot the significantly enriched pathways or gene ontology in bar graph, scatter plot, like showing on the right, and all the significance is indicated as with asterisks in the bar graph. And the scatter plots will also add another information to show you the counts of the differentially expressed transcripts. So again, as I mentioned, it will be performed for two different data sets. One will be the differentially expressed messenger RNAs, and the other will be the LincRNA targeting messenger RNAs. And for the CAG enrichment analysis, we will also include the significant CAG pathways in your report or in your release, in your result release package. And the last one I want to share will be the PPI, the protein-protein interaction. So you can input your data into the string database, and then through the database searching, we will get the potential interaction list output. So with that, we also perform that for two parts. One is for the messenger RNAs, and the other will be the interaction network between LincRNA and their target messenger RNAs. So the result will be something like this. Anything in a circle or a bubble will be one transcript. And if there is any connection between two bubbles, that indicates the potential interaction between those two transcripts. So after we finish all those analyses, we will run the data release. And in your data package, each separate folder will include all the publication-ready results for each of our analysis steps, as I indicated with those numbers. So you will find all those publication-ready results in each folder. And basically, our LincRNA services has been supported a lot of researchers to profile their either LincRNA or messenger RNAs, as indicated in this case. So here, they just ran a LincRNA sequencing for the mouse species, for the mouse samples. And in their result, they kind of showing up the differentially expressed LincRNAs and also messenger RNAs by using the heat maps. And also in the table, as I mentioned, we predicted some novel genes as indicated as the X log. And we also provide the information of their potential targets. So through the LincRNA services, it's quite easy for you to get a profile of your LincRNA and the messenger RNA networks in your samples or in your experimental designed species. So that will be everything I want to cover in the LincRNA part. And now let's check what kind of other solutions or non-coding RNA solutions we can provide for you by Novogene. So circRNA is also an important non-coding RNAs. It can function as a microRNA sponges to sponge the microRNA, and it will indirectly involved in the regulation as well. So their significance has been discovered in different diseases, especially in cancer, and served as a new diagnostics method for some different diseases. And to run the analysis, we also need the reference genome and complete annotation as well. And for the circRNA, as I mentioned, it must be the strand-specific library with ribosome removal. And the other option or the other treatment we will perform is the linear RNA removal for the circRNA, as indicated by their either name or configuration. So we will remove or clean up all the linear RNAs before we move forward to construct the library for the circRNAs. And here will be the data recommendation and the sample requirements. The other service we can provide for the non-coding RNA will be the microRNA sequencing. They have their special configuration in their sequences. at the both ends. And for this, you can just simply move forward with the library construction. And after we have the library ready and run a gel, you can just select based on their size. So it will be quite efficiently for us to construct the small RNA library directly. And again, for analysis, we need reference genome and annotation ready. As it only has the size between 18 to 40 nucleotides, we will use single-end sequencing, single-end 50 for the sequencing strategy. So you don't need to order the pair-end reads or even longer sequencing strategy for the small RNAs. And here will be the data recommendation, and below is the sample requirements. So through our individual non-coding RNA services, you may be able to study the LincRNA, messenger RNA network, circular or microRNA network, or even microRNA versus the messenger RNA network. So it gives you an idea about those three parts I have been shaded. So those will just give you kind of a potential network for only two types of RNAs. And this has been commonly used in a lot of researches, like the example I'm showing here. So in this review, it summarizes the LincRNA dysregulation in the cancer cells. As you can see here, through several years of study, they summarize or they kind of draw up or finish the whole picture of how my LincRNA involve in the cancer cells or cancer pathogenesis. So such as the LincRNA can sponge in microRNA, or they can interact with the protein, or they can interact with the messenger RNAs. So it takes years for us to fully draw this whole map or this whole picture. How can we get the global map more efficiently? Such as I really want to study not only the messenger RNA versus my microRNA, I also want to see the potential interaction between circRNA and the microRNA. You may think about maybe I can order those services individually or separately, but once you deliver different samples or different RNAs in different batches, somehow we may introduce some bias with no reason, such as I even collect my samples at the exactly same time. So I may even introduce some bias in my samples or in my data. So that's why we offer this service here, the whole transcriptome RNA-seq services. In this one, what you only need to do is you also deliver the total RNA to us. So you only need to send us your total RNA, and we don't need too much. We only need more than 5 micrograms. And through this input RNA, we will construct 2 different libraries. The first one will be the ribosome removal directional RNA library, and the other will be the small RNA library. So from this library, we'll get the data for LincRNA, messenger RNA, and the circRNA. And for the small RNA library, from small RNA library, we'll get the small RNA data. And with all the data ready from the same input sample, we can run the analysis to study the global map. So in our analysis, it will include 2 parts. First will be the individual standard analysis package. So again, this will be the same thing as I introduced in previous slides. And we will also include a free joint analysis to study the networks between even 3 types of RNAs. So I will mention the details for the joint analysis in future slides. Before we move on, I want to emphasize 2 points. First, this circular RNA library is not linear RNA removal library. So you may think that those information may be different if I use different library preparation. So the answer is quite possibly yes. But that really depends on your research objective. If you only want to study the circular RNA, and I will highly recommend you to use the linear RNA removal library with ribosome removal, with the directional library constructed. But if you really care about the network between all those multiple types of RNAs, so the ribosome removal and directional library will be enough to cover those informations. So it really depends on your research purpose. And the other point I want to emphasize is, if you order those services separately, you may need to prepare around 10 microgram of your total RNA. But if you check the input of our whole transcriptome RNA-seq service, it's just a half of it. So that's why we usually offer the whole transcriptome RNA-seq if you really want to study the network between multiple types of RNAs, or you have limited input samples. And now let's go check the details for our joint analysis. So in addition to the individual non-coding RNA-seq analysis as indicated in this pipeline, so finish the prediction of the targets, we will deliver the joint analysis as well. So the first joint analysis we will run will be the microRNA and the messenger RNA interaction. So we will check the differentially expressed messenger RNAs and their potential microRNA regulators. We'll also check the differentially expressed microRNAs, their potential targets. And then we cross-compare those two data sets and to get the overlap between those two results and run the enrichment analysis. So those will be the similar enrichment, like the co-enrichment and CAG enrichment. Through this, we constructed the microRNA and the messenger RNA regulatory or regulation network. And the second joint analysis will be the network between microRNA, messenger RNA, and LincRNA. So in this one, we will compare the LincRNA versus the microRNA precursors through the homology of their sequences. And we will also run the microRNA targets prediction to predict if there is any LincRNA is the microRNA targets. And then we compare those overlaps and also the overlap, also the prediction of the network from the microRNA and the messenger RNA, and select the network between those three types of the RNAs. So in this network construction, all three different RNAs are all differentially expressed after our analysis. And the third joint analysis is quite similar. So it's just a run very similar for the circRNA as well. So we compare the circRNA and check the source of their source messenger RNAs. And we also check the differentially circRNA versus some microRNAs. And then we cross-compare and run the selection and pick up the network between those three tabs. And also, those three tabs will be all differentially expressed in the network drawing or in the network results. So why we want to do that? As indicated in the figure showing on the right, so you can see those long non-coding RNAs or the circRNAs will both act as the microRNA sponges. So those can just attract or interact with those microRNAs and then change the expression level of those messenger RNAs or genes. So that's why we want to run those network between those three tabs or the three tabs. So here, I want to show another case using our whole transcriptome study. So in this one, we sequenced the whole transcriptome for their samples, and we identified more than 100 differentially expressed LincRNA and circRNA. Very few number, like for the differentially microRNA, and a lot of differentially messenger RNA, differentially expressed messenger RNAs. And by using our joint analysis through different steps or different network studies, we conclude with those networks to show their potential interaction between microRNA, genes, and lncRNA. And again, I want to emphasize, so I only show the lncRNA figure or result here, but they also include, or they also generate the results for the circular RNA network as well. So again, back to here. So even though today we only go through the introduction for the non-coding RNA services, we also have all other services available for you in Novogene. And if you need any joint analysis or customized analysis by combining different omics strategies, you can always contact us for the potential discussion for your project design. And to process all of those data, again, we have a very powerful processing system or calculating system. And with those system support, we have been support a lot of publications with a very high impact factors. So hopefully we can get involved in your next project and support you for your next publication. And with that, I'd like to thank you all again for your attention. And I'd like to take any questions.
So thank you, Whaley, for your very interesting presentation. So now let's start the Q&A session. So as some of you already have seen, some of the questions are already answered, and for the other, the experience of Whaley is needed. So unfortunately, we have only 10 minutes for this session. And if some question will not be answered, we will reply to you as soon as possible. So let's start from the first one. It's from Yuing Liu, who is asking that I have, sorry, I have a limited amount of total RNA. I know that you have a low input for messenger RNA. Do you also have related protocol for LincRNA?
So we always have the low input option for different types of RNAs, but the success, the success percentage, or how we can construct the library successfully, it really depends on your sample status and your input amount. So maybe our team can follow you up with your detailed sample information, like what type of samples you are having and how many total RNAs, how much total RNAs you can provide. And then we can work through those details and give you a finalized protocol for the low input library preparation.
Okay, thank you. And then Daniela Perovic is asking if the mapping rate of my sample is 80% circa, can this reference be used for further analysis?
This really depends on a lot of factors. So yes, for the reference genome, the mapping rate is a very important factor to check. But the other thing we need to confirm about of the reference set will be the annotation. So we really need a fully annotation to run the detailed analysis. And the other part is for your data. So as long as your data have a very universal mapping rate, like all of my samples will have a mapping rate around 80%. So that is a good sign for us to move forward. Or if there is like 2 out of 10 samples have a mapping rate as high as 99%, but all the other samples will have 80%, then we really need to double check those data and your sample to see where the problem is. So it really depends on on both sides, the reference side and also your data quality. So still it's kind of like a case by case question. So we also need to check the project in details.
Yeah, sure. Yeah. And then Sivakumar Krishnamurthy is asking why not we use TPM instead of FPKM?
Well, this is a quite interesting question. Thank you for this question. I have seen a lot of discussion between the TPM and versus the FPKM. So I will say it really doesn't matter which one you use because both of them are commonly used the normalization method for the RNA-seq data. And a lot of paper are still accepting the FPKM for their for the publication. So for us, our standard pipeline is using FPKM to show the expression levels in the quantification part. But if you need, we can also work together to see how we can create the TPM result for you. And the other thing I want to mention is, so the FPKM is just for us to visualize our data distribution, like the expression level distribution. So for the differential expression part, we usually don't use FPKM as input. Like for the messenger RNA-seq analysis, we use raw data because the software have their own normalization method to run the differential expression analysis. Well, for the other non-coding RNA-seq analysis, like the link, so we use different softwares. We follow the string type, and then we use either cuff compare or some other softwares. So they always have their output from the previous steps. So we don't just use the FPKM as the input to run the differential expression analysis. So again, back to your question, it's just a visualization of our sample distribution. So we don't use it for the differential expression analysis.
Okay, thank you. And then Zelika Trumbic is asking, what about if there is no reference genome available? Is it possible to do a de novo assembly?
De novo assembling pipeline is ready in Novogene, but only for the messenger RNA-seq. So for the messenger RNA-seq, we do have the de novo pipeline ready. But for the non-coding RNA services, it's quite hard for us to provide the non-coding RNA services. And we can also discuss this question based on your study research, your research objective and your project, because if there is no reference genome available, it may be possible to use some close relatives as a reference genome. So we can work through that for more details for this question.
Mm-hmm. Okay. Then Alexander Radanovic is asking, if Novogene is extracting RNA, what type of plant tissue you work with? And when analyzing long-encoding RNA, is mRNA analysis automatically included, or it has to be emazed separately?
So that's I will answer the first question first. So for the plant tissues, you can discuss the details of with our team. So we will work through the samples with you for more details. And basically for the plant, as far as I know, we accept multiple or different type of tissues. And there may be different treatments for different plants, such as some plants may have a lot of different chemicals. It's very hard to deal with. So we may use different kit, but for majority of the plants, it's quite possible for us to perform the extraction. And also for RNA-seq, as they have a very tissue-specific transcriptome in plants. So it really depends on your research objective. We may suggest you to provide a different type of tissues for extraction or for your projects. And for the second question, so you are asking if the messenger RNA is included in the LincRNA service or is a separate one. So the messenger RNA analysis part is included in the LincRNA. So you only need to order the LincRNA service, and we will deliver both LincRNA and the messenger RNA results to you. So you don't have to order them separately.
Okay, thank you. Sorry. And then Kostas Triantafilopoulos, I hope that I'm telling well the names, is asking, what type of tissue do you accept to proceed in-house with small RNA isolation inside Novogene?
You mean what type of
tissue? Yeah, for animal, plant. I think it's a general question regarding which kind of tissue we can accept. So if we have to do the RNA extraction.
Yeah, for the tissue thing, we basically accept or majority of the species, including plant, animal, or plant, animal, or microbial. So we kind of work with all of those species. And even we also have the meta-transcriptome services. So even you want to extract the RNA from some meta or some microbial community, or even soil or feces, so that is quite possible. So in general, we are accepting majority of those tissues or species, but again, so we need to confirm the details first before we give you a certain yes. Yeah.
He's asking more specifically, do you accept frozen blood?
For frozen blood, I think it's possible. I think we also have the instruction for the frozen blood delivery or sample preparation. I think our after the webinar, our either TS technical support team or our local representative will follow you up with the detailed instruction.
Yeah. Okay. Thank you. So I think that, yeah, I think that we don't have any more time to reply to the questions. So I would like to thank you, all of you, for your time today. We hope you find the webinar very useful for your project, your plan in your research. We will email you a copy of the slide deck shortly, and we will also send you the program for our future webinar. As John said, we will have a new one in July, and then we will stop in August to restart in September. And in the meantime, please stay in touch. You can follow us on Twitter, on LinkedIn, or you can get in touch via our website at en.novogene.com. So we look forward to see you all on the next webinar, and I wish you a good day. Thank you very much.